This is a recording for Biology 1407, Chapter 26, Seed Plants. So once again, we're taking a look at the phylogenetic tree right here, showing the plants of interest. We've already taken a look at the charophytes and the non-vascular plants, which is the bryophytes, which included three distinct groups, all collectively sharing the same life cycle details and similar adaptations to terrestrial life, including the embryophyte life cycle, where the diploid organism of the next generation is retained temporarily within the gametophyte of the previous generation for some development before it's released. In the case of the bryophytes, that diploid embryo matures into a sporophyte that releases spores to recapitulate the life cycle. A noticeable feature of those plants is the absence of vascular tissue and, by extension, structural tissue, which limits the size and complexity of these organisms because of their inability to overcome diffusion limitations associated with large size, as we've studied previously in animals. Then we took a look at vascular seedless plants, and that included the lycophytes and pterophytes right here, club mosses and all of their relations, and ferns and all of their relations right here for the pterophytes. And this is the group that you'd be more familiar with. The big innovation here is the development of vascular tissue, which is primarily expressed in the sporophyte generation. So the gametophytes in these organisms have become reduced, very, very small. Not particularly large or long-lived, and it is the sporophyte that is the plant that we primarily see when we're looking at these organisms right here. Whereas before in the seedless plants, the bryophytes, when you look at moss or a hornwort or a liverwort, you're normally looking at the gametophyte, and if you're there at the right time, you'll see the sporophytes growing up out of the mosses, liverworts, and hornworts as spore-releasing structures. They have a very short lifespan. Here it is the sporophytes that are the large, long-lived organism, and they're photosynthetic, of course. They have green parts and chlorophyll and all that. And the gametophyte is much reduced. It's a separate individual plant, but lives only long enough to produce its gametangia, its gametes, the gametes to fuse to form a new sporophyte, and then that's the thing again that you're looking at. A fern is a sporophyte organism, as is a club moss or a spike moss. The vascular tissue and associated structural tissue overcomes diffusion distance limitations and allows for the growth of very, very large organisms. Modern day ferns, there are some tree ferns you can find in some tropical locations, but in the Mesozoic era, tree ferns would have been one of the dominant plant forms on Earth, and there were some really spectacularly large ones in the fossil record. Interestingly, a heck of a lot of the coal that we have been using for the last century includes fossils of a lot of ferns and various other members of the lycophyte and pterophyte clade of terrestrial plants, and some early gymnosperms show up in that fossil record as well, of fossilized organic material in the form of coal. So on to seed plants, or the spermatophytes. And the seed plants are characterized primarily by the development of this seed structure. So they're vascular, they're embryophytes, they are green plants with the chlorophyll and various other molecular structures that are shared amongst all of these organisms. But the big innovation is the production of a seed, which is a means of dispersal that combines the delivery of gametes from male to female, or alters the mechanism for the delivery of gametes from male to female, and combines the distribution of new organisms, which used to be done by delivering spores, releasing spores into the environment. All of these organisms release spores as their means of colonizing new areas. The spore and reproduction have been combined together in the seed plants in the form of the seed. So we'll see sporopollen, which was a feature surrounding the spores of these plants right here. That compound is retained, but it surrounds a new thing called a pollen structure, which we'll talk about, which is a characteristic of seed plants. First, we'll take a look at gymnosperms, and then we'll take a look at angiosperms. And remember, another synonym for vascular plants is tracheophytes, because they contain these specialized cells called tracheids. All right. So there's a lot of ambiguity in the fossil record, but up to 465 million years ago, we see the first evidence in the fossil record of seed-bearing plants, and those would have been the gymnosperms. The angiosperms were a later arrival that evolved from gymnosperms. And the reason these plants are so successful, about 1,000 species of gymnosperms and about 300 species of angiosperms or flowering plants, is that seed right here. And the seed satisfies a number of different functions. For one, as it says right here, it protects and provides food for the embryo. So the embryo is the new diploid plant. The new little diploid plant inside the seed is the embryo. It's surrounded in a seed by some material, sacrificial material, that provides food in the form of stored metabolites, carbohydrates, and lipids for that embryo. And then all of that is wrapped in some sort of protective, covering right here. So a seed is a complicated structure consisting of an embryo, food, and an outer protective covering. Another innovation, which is shared a little bit with the spores, is the ability, but this is much amplified, dormancy. So the idea is that, seeds of a lot of plants have a built-in timer, if you will, that may hold development, germination for an extended period of time, until the environment becomes more benign or more hospitable to germination of that seed. So the seed is able to, A, be put into a state of suspended animation, sort of an internal timer, and it is sensitive to conditions of its environment. So there are means by which environmental variables, light, water, presence of nutrients would be the key factors right here. Sometimes temperature will induce or inhibit germination of the seed. So the seed can selectively know when to actually germinate and produce the next generation of plants. So that's a really useful thing. In the case of flowering plants, the development of fruits enhance seed dispersal through manipulation of animals to move those seeds about by eating or storing or moving fruit from one location to another. Finally, reproduction, one of the great innovations of the seed plants, so all of the gymnosperms and angiosperms, is pollen. So pollen is a structure produced by the sporophyte portion of a pine cone or a flower on a plant, and that's all sporophyte tissue. And pollen contains within it a male gametophyte, so a multicellular little haploid plant. It consists of just a few cells, and delivers the sperm cell, a product of a gametophyte that has never changed, to ultimately a female gametophyte. And so now, for the first time, sperm do not have to swim through water to get to an egg. They are delivered via other mechanisms through the structure, this complicated structure called a pollen grain or pollen structure. And again, animals figure prominently, especially in the world of angiosperms, in the movement of this pollen. All right, a little bit about a seed right here. So we're looking at a bean right here. This could be a pinto bean or a brown bean or something like that. And couple of things right here. Here's the embryo. So if you take a bean that's been wetted, say if you take a bean at home and soak it in water for a little while until something starts happening, about a day is usually plenty, or half a day even, what you'll notice is that the embryo is reanimating. So that just tells you, even a dried, seed that we're keeping at home as food really is in suspended animation and can reanimate when exposed to water very, very clearly. And if once it's started cracking open on its own, if you very carefully dissect it, you'll be able to easily see the embryo. It's going to be pale white yellow until it's exposed to light. Once it cracks open and it's exposed to light, you'll see the expression of genes coming on that ultimately lead to the production of chlorophyll. So you'll see the chlorophyll, which lets you know that a bunch of genes coding for various metabolite production are coming on. Anyway, that's the embryo right there. And then there's a terrific amount of material scattered about all over the place right here. I'm sorry, I'm not pointing enough. The embryo includes these two what are called seed leaves right here, the cotyledons. And then an extended, a larger region right here. This is actually parts of the cotyledons in the case of the bean right here. They're kind of unusual seeds. But this big structure, the two halves that we eat when we're eating the bean, are full of protein and oils and a little bit of carbohydrates. And that is the food material to nourish the young embryo when it's first developing. Then on the outside, we have this integument right here, color-coded in brown on the slide. Which hardens into a seed coat, and that is sporophyte tissue. So that's tissue of the parent plant. Here's the new diploid embryo in here, and then we've got tissue that's derived from the gametophyte in here. We'll talk about that later. Another thing that we see consistently in seed plants, and it appears in some of the seedless plants. Some of the ferns exhibit this behavior too, but where there are two types of spores and two types of gametophytes. And so what happens is there is a microspore, that is a very, very tiny spore cell that is produced, that produces a very, very small male gametophyte, and that's called a microgametophyte. And that microgametophyte is going to mature into ultimately this pollen structure, this pollen grain. And then there is another type of spore, a large spore, which germinates into a female gametophyte, and that is going to be a larger structure, more cells involved. But there are two different types of spores right here, so we have two different genders of these structures. And that female gametophyte is going to develop into a structure called an ovule right here, which should, let you know that there's an egg involved in here somewhere. Ovum is, ovary, and all of those terms are familiar to you. So this has to do with egg production. And that ovule is also going to be contained within the parental sporophyte, the large plant that we normally look at and think about. All right. So there's some groups to consider here. Most importantly for y'all are to be able to talk about the shared characteristics of all gymnosperms. So I'm not going to test you literally on these terms right here, but you do need to, just for the record, there's a number of different major groups right here. Starting at the bottom, the Ginkgophyta is a ancient group of gymnosperms that very few exist today. Really, only one species and a couple of variants of that species called the ginkgo. This is a decorative tree. We have them on our campus at TCC, Ginkgo biloba. If you're into nutrition and personal fitness and health and stuff like that, and visit nutrition stores and stuff like that, you'll be familiar with the nutritional supplement ginkgo. Which is thought to have beneficial properties for cognition and memory and things like that. That's taken from the ginkgo tree, which is a gymnosperm. It produces a type of cone. It looks like a little small soccer ball on the female plants, and a very, very distinct characteristic leaf of the ginkgo. The cnidophytes right here are another very interesting group of kind of old relic gymnosperms. There's not many species around today. The most famous one is Welwitschia, which is a very, very exotic looking and very, very unique plant that grows in extreme desert environments in southwest Africa, Namibia to be specific. Welwitschia, and that is a member of the cnidophyta group. The cycads are more familiar to people. These are used a lot as a decorative plant. They look vaguely like a fern, but if you touch them, they're very, very tough and hard and heavily sclerotized stems and leaves. and they only superficially look like a fern. They're actually a cone-bearing plant. The cones are born in the center of the rosette of leaves. And then finally the coniferophyta, which is the pine trees and juniper trees and cypress trees and all of the others. This is by far the most diverse group of surviving plants today. And then, easy for y'all, there's only one group of angiosperms, the Anthophyta right here, so that collectively includes all the angiosperms. There are divisions within here which are of interest as well, but the angiosperms are all of the flowering plants. All right. So what does gymnosperm mean? It actually means naked seeds. So that's the meaning of that term right there when you translate the Greek roots. Naked seeds. So these are seeds that are not protected by a secondary structure, which is what in flowering plants would be the fruit. The fruit is the covering around the seed that's not present in the gymnosperms. All right. So again, there are these four living groups right here, and many ancient extinct groups that don't exist anymore. And the ginkgophytes are really considered a living fossil, and the gonidophytes, or the netophytes, I'm sorry, would be kind of cast in that same light as being living fossils, if you will. So what they have is a structure called a cone, and there's a lot of variation in this cone structure, but all of the cones are characterized by a thing called a scale. So when you visualize a pine cone in your mind, and we'll look at a picture in a bit, each of the individual pieces of that cone is called a scale. It's kind of like a leaf. It is, in fact, a modified leaf. And on that scale is going to be an ovule, an exposed ovule on that scale, which is, again, part of the reason for calling these naked seeds, because that ovule is exposed and visible at some point in the development of the cone. All right, so here's some cones right here on a pine tree. And then some micrographs, microscopic views of these things right here. All right, and gosh darn it, this is a slide prepared by OpenStax, and it doesn't have magnification on here, so very bad of them. There should be a magnification statement right beside these photographs. Don't y'all copy that. All right, that habit of forgetting the magnification. So here's a pine tree. And if you weren't aware of it, pines have gender. So there is such a thing as a male tree, there is such a thing as a female tree, and then for a lot of species, they are both male and female. So the sporophyte can be, what's the word? Hermaphroditic, that it has structures of both genders as far as reproduction goes. Anyway, the two types of reproductive structure for the female is a female cone, which is very large, and this picture doesn't really do justice, and then the male cone, which is much smaller and much more delicate and powdery, and that's going to be pollen. When you see a male cone, you're looking at a male cone if it's really small, oftentimes kind of this golden color right here, and if it's mature, if you touch it, dust comes off of it, and that's pollen. The female cone is inevitably more robust, more structurally substantial, usually much, much larger than the male cone. So, this picture doesn't really capture the difference in sizes as well as it should, but for the purposes of illustration, it's great. And so within that cone, here's a female cone, here's a male cone down here. On the female cone, we've got a modified leaf called a scale, and on there is an ovule, which is a compartment where, and this is what it is a sporangium, just like on our ferns, just like back on the sporophyte of a moss even, a sporangium, so a capsule that produces haploid spores via meiosis, so all of that occurs down in here. But then what happens is the haploid spore, and there's only one of them, or in the case of these gymnosperms, two. There are two specifically, but that's a detail that's unimportant right now. But the spore then germinates in place. So the female gametophyte germinates inside the ovule on the female sporophyte. So that's very different. When we looked at a fern, the female gametophyte actually germinates on the ground as a excuse me, as a distinct little gametophyte, and there's no gender. They're both male and female at the same time. And in a moss, the gametophyte is, the moss that we're looking at. But here, the gametophyte is retained within this tiny little capsule right here, and matures completely in here, and ultimately produces an egg. So, what do gametophytes produce? Gametophytes are plants that produce gametes. So the female gametophyte in this little capsule right here produces an egg. Similar thing is happening on the male cone right here. We've got structures on the scales that are sporangia, but these are microsporangia, meaning they produce tiny spores, and they produce lots and lots of microsporangia. And those microsporangia, that are produced, those microspores that are produced by the microsporangia, they're produced via meiosis again, so they're haploid cells within a diploid structure. The cone is diploid. The sporangia are diploid, but the cells in them, the spores, are haploid. These spores will also germinate, so they will turn into multicellular little tiny male gametophytes, and they will mature into a structure called a pollen grain and ultimately be released. So that's the powder that you'll see on the surface of this mature male cone, is the male gametophyte surrounded by a layer of sporopollen. And by the way, the sporopollen comes from the diploid parent. So a pollen grain is diploid tissue in the form of a protein surrounding a male gametophyte that is haploid and containing male gametes that are haploid. That's what a pollen grain is. Sporophyte on the outside, male gametophyte on the inside. All right, and so here's some examples of the conifers right here. Here's a coastal redwood, coastal redwoods, and what am I trying to say? And I can't think of their names. Sequoias, giant sequoias, are the biggest living conifers. The tallest plants on Earth, about 300 feet tall, a little over 100 meters tall, quite spectacular. The oldest known plants, single plants on Earth, some bristlecone pines. Some of these guys are, tens of thousands of years old, very slow growing bristlecone pines. So the oldest conifers and the biggest conifers on the planet are both in North America. So we're kind of lucky here in America to have these things, the tallest and the biggest and the oldest plants on Earth, all on North America. The bristlecone pine is famous. All right. So let's go ahead and take a look at this little video right here. All right, let's see how this works.
A pine is a common conifer found all over the northern hemisphere. It can be used as a model for the seed production process in all gymnosperms. The adult sporophyte, the pine tree, develops male and female cones on separate branches. Female cones develop 2 ovules on the upper surface of each cone scale. Each ovule contains haploid megaspores. Male cones produce microspores by meiosis. The microspores develop into pollen grains. Each winged pollen grain is a 4-celled male gametophyte.
So one thing here, we're seeing 4 pollen grains being shown developing. That's to kind of highlight the fact that the microspore cell, so the male the sporangium, the male sporangium produces microspore cells that divide via meiosis to produce 4 haploid microspores, and then those 4 haploid microspores germinate to become a multicellular male gametophyte, and that's going to be ultimately the pollen grain. They didn't linger on it, but in the female megasporangium on this female cone of a sporophyte, the female megasporangium, which is still a sporophyte structure, that means diploid, produces a megaspore cell that divides. It's called a megaspore mother cell, in case you read that. It just means that's the diploid cell. And that cell also divides via meiosis, producing 4 cells. So here's the 4. But like in animals, and we didn't study animal reproduction, it's a pity, we just don't have time, but this is true for us as well. In the production of eggs, meiosis leads to 3 non-viable cells that are small and 1 big cell that retains all the cytoplasm. So that's unequal cytokinesis in meiosis, so that we get 1 really big ovum, egg. And so that's the megaspore right here, maturing into, this will, what's the word? This will germinate into the megaspore. So there's some interesting terminology here. All right, let's go back. Here we go. Yeah, pollen grains.
The female gametophyte grows, producing 2 or more archegonia, each of which contains an egg. During pollination, a wind-borne pollen grain falls near the opening in one of the ovules of the female cone. Each male gametophyte forms a pollen tube that penetrates the tissue of the female gametophyte. When the pollen tube has grown into the archegonium, a sperm cell from the male gametophyte fertilizes the egg. The zygote develops into an embryo and a mature seed is produced. The female cone opens, releasing the seeds. When conditions are favorable, the seed germinates into a new, young sporophyte, a pine tree seedling. Near the opening in one of the ovules.
So here's an interesting little thing. If you're looking at pollen and your pollen has wings, these are thin little wing-like things, and this is a large spherical central section right here, and kind of vaguely looks like a Mickey Mouse, cartoon, mouse head or something like that. That's gymnosperm. That is super characteristic of gymnosperms, of pines and all of their relations. Flowering plants produce pollen as well, but they never have the 2 ears. So that's an easy way of distinguishing the two. So just a little tidbit right there. So let's go back to this germination right here. Yeah, so here's the seed germinating
into a new, young sporophyte, a pine tree seedling.
All right, so hopefully that was a little bit useful. So that, captures the key events in the reproduction of a gymnosperm. So remember, there is a microgametophyte that is contained within a pollen grain. That is, the core of the pollen grain. And there is a megagametophyte, which produces an ovule. These contain sperm cells. These contain eggs. And that's the means of reproduction in the sporophyte, in the gymnosperms. All right, here's a cycad. So these have the same life cycle. A cycad, as you can see, superficially looks a little bit like a fern, but you'll find that these are not fronds. They don't uncurl, and they don't have spore-producing structures ever on the underside. This is a small tree fern right here. It has a trunk. So back in the age of dinosaurs, let's say 50, 60s, 70, 80, 90 million years ago, 100 million years ago, very large versions of this plant were to be found and would have been food for herbivorous dinosaurs, oftentimes used as that. And at the right time of year, we'll see cones in the center right here. These are going to be female cones right here. They're very large, very, very structurally substantial. The male cones will be smaller, and that's going to be over on a male plant. So these are male cones right here on female cones on a female cycad right here, and you'd have the equivalent looking situation on a male cycad. Here's the cnidophytes right here. This is ephedra, which grows on our continent right here, oftentimes called Mormon tea. This is the source of the stimulant ephedrine, which is used in certain types of antihistamines. So this grows in the desert southwest of the U.S. You can find it in Utah, particularly abundantly, northern New Mexico, Arizona, places like that. Netum, which is an unusual, rare, tropical gymnosperm that looks superficially like some flowering plants. And here's the famous Welwitschia of Namibia, the southwest coast of Africa. This is a desert that reaches right to the Atlantic coast pretty much. Super dry spot on the planet. kind of the African equivalent of the dry plains of Peru and northern Chile. Same sort of climatic conditions cause that desert. And this plant right here is a famous, long-lived, very, very unusual organism. There's a great video on Welwitschia. It's not core to this unit right here, so I'll leave this for you to watch, but I strongly encourage it. If you're curious, go ahead and watch this video. The ginkgos right here. Whenever we get back on campus, look for the ginkgo trees that are between the life sciences buildings and the library. There's a couple of them, and they're very easily identified by this parasol-shaped leaf right here. Looks kind of like a fan, not parasol, sorry, fan-shaped leaf. And the females produce a cone that looks like a little soccer ball. So you know you're looking at a conifer when you see that cone-like structure. But instead of looking like a pine cone, it looks like a strange little ball sphere made out of repeating fragments. Very interesting. All right. The last thing we want to look at in here is the angiosperms. And of course, the key characteristic that distinguishes angiosperms from gymnosperms, one of two, is the production of flowers. The other big modification is the production of fruit. They don't have naked seeds. They have protected seeds that are protected in a structure called a fruit. If you're really wanting to study botany, there's going to be a lot of divisions within the angiosperms, not to be an intimidating term, but just there's lots of interesting subdivisions. There are two, though, for the general student that you do want to be familiar with, because you'll hear about these a lot, and that's the monocots and the dicots, specifically referred to as the U or true dicots, which implies there's some false dicots out there as well. Unimportant right now. Monocots and dicots, however, are two particular forms that flowering plants take that are extremely important and well represented around the world today. All right, so let's go ahead and talk about those a little bit. So monocots, the word is an abbreviation of monocotyledon and dicotyledon, and a cotyledon is the first leaf that emerges from a seed. We're familiar with that from our work in laboratory, looking at the fast plants, the Brassica rapa, the mustard, the wild mustard. And when those seeds germinated, they had two initial very distinct leaves, kind of big, round, paddle-shaped, fat-bodied leaves that were very different from all the rest of the leaves on the plant, the ones further up on the stem, and those are the cotyledons. And so in dicots, you get two of those. In monocots, you get one. So that's where the name comes from, and that's the key feature. There's one seed leaf in monocots. That'd be corn and grasses and wheat and things like that. And two seed leaves in a eudicot. Think of the two seed leaves on a bean after it first germinates, which hopefully some of y'all have seen. Other structures that are pretty consistent right here, when we look at the venation, the vascular tissue in the leaves, they're in parallel rows in monocots. They are branched, inevitably branching, sometimes quite complicated branching patterns in dicots. When we look at the vascular tissues in the stem, if we cut a stem across, we see vascular bundles, which consist of xylem and phloem, scattered about somewhat randomly throughout the stem. In a dicot, they're inevitably arranged in this ring pattern, a very, very distinct, very organized ring pattern. The roots, think about when you pull grass up out of the ground, you get just a pile of roots. It's called a network of fibrous roots. There's no one main root. There's just a whole lot of roots that all seem to emanate from the same point. On the other hand, dicots consistently have a central deep root. Think about the weeds you were trying to pull out of your lawn if you're one of those people that hates weeds. Dandelion would be a great example. If you don't get the taproot, the plant could even come back sometimes. So the taproot is a distinct stem, if you will, running underground with branches, lateral branches off to the sides coming off of it, a very different model. If you're looking at pollen, under the microscope. This is really important in a lot of paleontological work, where we're looking at old fossilized material, because pollen fossilizes really well. We can learn a lot about the ecology of an area by looking at fossilized pollen. Anyway, the pollen grains in monocots have a single little line, a kind of a groove cut into them, monosulcate. The sulca is a groove or dent. So there's one distinct one on monocots, and dicots have three of them. So it's real consistent. So if you see pollen, that's been dug up out of out of some deposit somewhere, some fossilized deposit. If it's got ears, it's a gymnosperm, pure and simple. If it looks like a Mickey Mouse head. If it's spherical but with a single groove in it, it's going to be a monocot. If it's spherical with three grooves in it, it's going to be a dicot. And then finally, the flowering parts, and y'all can check this anytime you want. If you go out and look at a flower, and it has three, or real distinctly, obviously and clearly, multiples of three petals and various other parts, pretty sure it's going to be a monocot. Think of a lily. Think of any number of other monocot flowers. There's another big group that's escaping me right now, but we have a bunch of them growing on campus. In the dicots, the flowers inevitably have 4 or 5 part symmetries. So 4 petals, or 8 petals, or 12, or 16, or things like that, or 5, 10, 15, 20, 25 petals. An apple, for instance. The apple flower very distinctly has 5 very, very clear, obvious petals. So examples. And same thing for the other parts of a flower. We'll see there's several parts to a flower. So here is the angiosperm life cycle, and you need to be able to do this. Your homework assignment requires you to do a life cycle for a algae, like Chara, for a moss, for a fern, for a pine tree, and for a flowering plant. So here's the life cycle for a flowering plant. So we still have male and female. So when you look at a flower, and a lot of flowers are going to be hermaphroditic. I was talking about that with the pine trees, and that means that they have female parts, which are in the center, inevitably in the center, and male parts, which are going to be in the first whorl around the center. So this is, around the female part. Now there are such things as male flowers, so they'll only have these pieces right here, and there is no female portion that's absent. And there are such things as female-only flowers that don't have male parts. It just depends on the species. So recognize all 3 possibilities exist out there. Male flowers, female flowers, and more commonly, hermaphroditic flowers that are both male and female. In the case of those hermaphrodites, inevitably there's some sort of biochemical mechanism to inhibit self-pollination. It could still occur, but it occurs rarely or infrequently, because self-pollination would negate all the benefits of sexual reproduction. So on the male parts of the flower right here, we have a sporangium. So remember the flower itself, the plant itself. This is true for the angiosperms. The plant that you're looking at, including the flower, is a sporophyte. It's a diploid plant, sporophyte. But within that sporophyte, including in these portions right here, we have a structure called a sporangium. So we remember that all the way back from the mosses, from the bryophytes. And the sporangium is a diploid structure that ultimately will produce spores. So there's a spore mother cell, it's a diploid cell. It goes through meiotic division, meiosis, to produce 4 haploid cells, 1, 2, 3, 4. Each of those germinates into a pollen grain, which is a microgametophyte. That's a male pollen right here. It's a haploid cell because it's produced via mitosis. And it's a haploid cell right here. It germinates into a haploid organism. And there are generative cells, which are going to produce sperm. And then there's a tube cell, which will grow the structure that allows for fertilization. And collectively, that's the microgametophyte. It's surrounded by a sporopollen shell that is derived from the parent plant. So the sporopollen shell is technically sporophyte, diploid tissue around the outside right here, but inside is a haploid gametophyte. producing a gamete. The generative cell will be the gamete. And that's our pollen grain. On a female portion of a flower, we have a compartment called an ovary. This is all sporophytic diploid tissue. And within that ovary, we're going to have ovules, a sporophytic structure right here, where we have a megaspore cell, macrospore in this case, mega, macro, same thing. The macrospore divides via meiosis to produce 4 cells. All right. Only one of those cells will be retained it divides via meiosis to produce 4 cells. Those cells in turn go on through some additional divisions via mitosis to produce what's characteristically an 8-cell female gametophyte. So meiosis produced these haploid cells. Those haploid cells then divide via mitosis to produce a bunch more cells. And we get an egg. There's the ovum itself, the egg, and then a whole bunch of other cells that are going to contribute to seed development. And that's where we are until we have fertilization, however that occurs. And fertilization requires that a pollen grain make contact with a female flower or the female part of a hermaphroditic flower, and it's going to attach to the sticky surface right here of the stigma, which is covered in a sticky, in the case of flowering plants, sweet material. Sticky so it hangs onto the pollen, sweet so it attracts birds to move pollen from one flower to another, or insects, or whatever it may be, bats. And then that pollen grain, when it makes contact with the stigma, starts growing a pollen tube. There's a nucleus that drives the growth of that pollen tube, and that pollen tube reaches all the way into the ovary and will make contact with an ovule. And when it makes contact with an ovule, a generative cell is transmitted via that tube nucleus and fertilizes the egg right there. So that's the key event. There's going to be a second sperm nucleus also introduced in flowering plants, and that's going to fuse with these other nuclei in here and produce a triploid tissue called endosperm, which is characteristic of flowering plants, of angiosperms, and that endosperm will result in the protein and or carbohydrate and or lipid-rich material that is food for the embryo in the seed, the endosperm right here. So here's a seed that is produced via fertilization of an egg. So here's a seed, that's the ovum once it's matured. The outer coating of the ovule becomes the seed coat, a very, very tough, hard, desiccation-resistant diploid tissue from the sporophyte. Inside there is the remains of the female gametophyte, which has been converted into this triploid endosperm. So it's actually triploid. And then there is a new diploid embryo, the future plant, in the center right here, which will be nourished from this endosperm for some period of time. Here's an illustration of monocots versus dicots again. The list that we looked at previously right here, but now in visual form. So when we look at the flowers, three-part symmetry right here, 3 major petals, 3 minor petals right here for the monocot. Vascular bundles in the stems scattered about randomly. Long, narrow, straight leaves with parallel veins. In the dicots, 2 cotyledons versus 1 cotyledon over here. Branching, vein patterns on leaves, vascular bundles in a ring, and flower parts, in this case 1, 2, 3, 4, 5, 5-part symmetry. So a little bit about those monocots and dicots. What are they? Well, the monocots, for one, don't produce true wood. And the reason we say true wood is because there's false wood, and false wood would be the hard stuff that bamboo plants produce, or the tough stems, trunks of, say, a palm tree. A palm tree is a monocot. It's not true wood in the sense that it lacks some of the features that, say, a pine tree or an apple tree have. Okay, so there are very, very tough stem-like materials, but they're just not the same as the tough stem-like material on dicots. And those monocots include grasses and palm trees, therefore the palm trunk. Grasses, if you're not familiar with it, bamboo is the biggest and most spectacular example of a grass. The grasses that are more important to us are going to be a number of our foods. Our staple foods include rice, corn, and wheat, all three of which are grasses, and therefore monocots. Grass doesn't simply mean, the stuff on your lawn, but it refers to a whole big group of plants that have long, slim, parallel veined leaves, green stems. They're herbaceous. They never produce, most of them don't produce any kind of non-green material, which is what wood is, but some of them produce a false wood, so that's the trunk of a palm or the core of a bamboo stalk. And that's what we mean by herbaceous right here. In the world of eudicots, a lot of them do form true wood, not all. Mustard doesn't produce wood. It remains herbaceous, which just means it's green with a green stem. But a lot of eudicots do indeed produce true wood, as do gymnosperms. The conifers, pines and junipers and all of those guys produce their version of true wood as well. And what it clearly shows us is that flowering plants are descended from gymnosperms, and the dicots are actually the older group of flowering plants. They're more closely structurally related to their ancestors, the gymnosperms, while the monocots are a much more recent, highly derived group of angiosperms that have some unique features. They're much younger in the fossil record. So eudicots do produce true wood, as it says right here. For those that do produce true wood, they've got this thing called secondary growth, which is, instead of just growing from their tips, from the apical meristem, which is characteristic of all plants, the growing tip of the shoot, the growing tip of the stem, the growing tip of the branch, the growing tip of the root, all of that stuff. That's what we mean by an apical meristem. Secondary growth allows the stem to get wider and the main roots to get wider, to get bigger and larger in diameter. That's what secondary growth is. And in true wood, what we have ultimately is an accumulation of lots and lots of xylem. A huge amount of xylem is accumulated in secondary growth. So when you chop down a tree and look at the trunk, you're primarily looking at a bunch of xylem. The bark includes phloem and outer protective tissues. So here's a cross-section of the stem of, this is classically a basswood tree. This is the slide that we would typically have available in a laboratory teaching this material. And we've got the pith right here, which is kind of equivalent to the stem of a herbaceous plant. But then we have these growth rings, which are characteristic of dicots and conifers. Mouse isn't working quite correctly. Dicots and conifers, and these are these growth rings right here, which are very, very characteristic of both dichotomous flowering plants and gymnosperm, like a pine tree. And that these are these rings right here, these growth rings. And all of this is xylem, 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 xylem. Only the outer bits of xylem are actually functioning in distributing water and minerals around the plant. The inner xylem is really just dead and preserved. And then phloem on the outside, phloem out here, distributing sugars and metabolites, and then the various protective tissues that collectively make up the bark ring around this. So that's very characteristic of a woody dicot. All right, that concludes the lecture on seed plants, the second of the 4 chapters in this particular unit.